¿Tú?
Escucha, Susana, tú lo das en normal y corriente, ¿no?
Everything in English then. Okay. I'm waiting. I can start. Okay. Okay. Good morning, everybody. Welcome to this new talk of Friday afternoon. It is my pleasure to introduce today's speaker, Susanna Manrubia. For those that you haven't seen her around, she's been here since December 24, so it's more than a year. And we've been trying to get her to give this talk for a year then, more or less. But basically that she's a very busy professor of investigation, and that's why we have been unable to get to this point till today. So I have this in Spanish. Okay, I will make a translation right away. So Susana studied physics sciences in the Universidad de Barcelona and got the PhD degree in the Universidad Politécnica de Catalunya. And then she moved to several Max Planck institutes in Germany for a while, and then returned and then make also a nice trip around the CSIC. She has been in the Centro de Astrobiología, INTA, CSIC, for more than a decade. Then CNB, Centro Nacional de Biotenología, where then she became deputy director of the Centro Nacional de Biotenología. And then, for the moment, she's here, seeing that she's like an electron that moves around a lot and spin-offs. For the moment, we are glad to have her here around. And one of the things that she's an expert in translating this, in theoretical models, computational and theoretical models of evolution. That's why she's around, and she wanted to be here at the population level, molecular level, and is basically using her knowledge, physical knowledge, into work on quantitative analysis of adaptive and evolutionary mechanisms. And then she's gonna briefly explain a little bit more or not. And basically, just to finish my introduction, she's been in the Agencia Agencia Española de Investigación, and has more than 100 papers and several projects from the Plan Nacional. And I would like to highlight here that one of the things that we talked about when she was coming is that she's very interested in outreach. And so here's a place in the museum that, he can fulfill that necessity by working in exhibitions, educational programs, and so on. And she has a nice book called Genes y Genealogías sobre nuestra herencia cultural y biológica. And I stop here my introduction. I pass the floor to her.
Thank you. Well, thank you very much. I first want to thank you all for coming, despite the weather, despite the trains, despite the Friday, and so on. Yeah. So I'm very glad to see the room so full, despite these elements. Okay. So I thought I was going to take this opportunity not to talk only about what the current projects we are developing here nowadays, but also to sort of explain why I am here. Because I guess that for many of you, physics is not something that is so directly, or a specialist in physics is not so directly related to the natural sciences, the kind of field work that many of you do, and the kind of approaches you use to describe your systems. So I'd like to show you otherwise, because there is a deep motivation behind what I have developed along my career. And I'd like to start with some introduction of what we physicists call complex systems. So this is murmuration of starlings, right? Which is a very poetic way of describing the behavior of these birds that I'm sure you're very familiar with, that they form all these shapes in when they fly, this group behavior, right? But it's clear that there is no plan here. There is no one directing what starlings should do when they fly together. And despite that, you see this kind of organization, or we call it self-organization of the patterns, of the displays they make, right? So this is another image. And so the key idea here is that you have an emerging behavior, collective behavior, due to the interaction of simple, quotation marks, simple elements, which are those birds. So this is very much how many problems in physics have been described through the history of physics, right? You have particles, elements, agents, wherever, that collectively have a behavior that transcends the individual, right? So more on that now in a few moments. So there are lots of systems that physics has tackled in the last, what can I say, 50 years or so, where these properties, these simple elements behaving collectively in a complex way are seen. So you have these stadiums where you have people after a concert clapping all together, this kind of synchronization. There is no one saying you have to clap at this rate. But despite that, there is this emergence of this clapping together. You have the universe and the gravitational interaction among galaxies at larger scales, and you have a formation of a fractal pattern where matter accumulates along sheets and, forming this kind of a structure. You also see it in many human collective behavior, like, for instance, in the formation of cities. Cities follow well-defined patterns that can be described through very simple mechanisms. So patterns in animals or chemical reactions, or of course, ants and termites, social insects. When you have these constructions of these animals, there is no ant that has a plan. Yeah? There is no architect ant saying we have to behave in this way. But despite that, you see the emergence of a collective behavior, right? And well, here are other examples, also in economics and so on. So this kind of physics that looks at patterns and processes in other areas and tries to contribute a view of the collective behavior of those systems, this is what we at large call complex systems, right? So the complex systems in physics essentially have two main historical contributions. The first one is chaos. I don't know if you have heard, not about the TV series, but about the problem, the three-body problem, right? So the idea is that when you have Newton's law, and you have the sun, and you have the planets orbiting around the sun, right? If you have just two-body problems, you can solve it exactly, and you have these elliptical orbits around the sun, right? But if you put a third body, Jupiter, then the dynamics are completely different, are chaotic. Chaotic in a sense, this is a simulation of three-body problems with three bodies of equal mass. This is the center of mass of the three bodies. And if you just look at the dynamics through the simple gravitational interaction of the three bodies, this is the kind of trajectories you see. So there is no periodicity here. There is no divergence. They don't escape somewhere out of where the system is confined, but there is no repetition of the behavior. So this is a complex dynamical system that, despite being deterministic, cannot be predicted because you cannot know with infinite precision neither the conditions of the system, neither the parameters that describe the dynamics. Right? You may be more familiar, perhaps, with the later, when we had the boom of chaos, was due to this person, Edward Lorenz, who had this system describing the convection, atmospheric convection. Just very simplified model with three equations, but the dynamics, the trajectories in the x, y, z space looked also chaotic, are also chaotic, right? So this means you start with, this is for one of the variables, for y, you start with initial conditions that are very close one to another, and after a certain time, you see a divergence of the trajectories, and it's impossible to predict. So this is the essence of our lack of ability to predict the atmospheric weather, for instance, because the equations describing the weather are chaotic. So there is a time, a typical time, when given some initial conditions and some parameters, you lose your ability to say what's going to happen afterwards. So don't blame so the weatherman, the weather person, because there is something intrinsic in this behavior, right? So this is what is known as the butterfly effect that you may have heard about. The other part of physics that contributes to these complex systems is statistical mechanics. Statistical mechanics is a very clear example of particles interacting and descriptions of this interaction in terms of macro variables. So you have a gas, you have the particles, but you look at temperature. Temperature is an emerging property of this system, right? Or pressure. You don't mind about what every individual particle is doing, what's the speed or how it's moving, where is it at a certain time, but what matters to you is the pressure and the temperature of this gas. And this is a good description of your system. So keep this idea in mind, because this is also how we sometimes address biological systems. We look at collective behaviors, at high-level descriptions of those systems, and we often do not care about what's going on with the specific particles. You see here, I don't mind if the particle is water or methane or something different. I don't mind which are the microscopic details. I can describe my system by macroscopic variables. Then here you have all these, well, these are the fathers of statistical mechanics, and you have some behaviors that you also describe, like different phases of the same system, or you have transitions, phase transitions among different states. So complex systems started, as I was saying, about half a century ago, but was not also very welcomed by physicists themselves, because this doesn't look serious physics for a physicist, right? You're worrying about what starlings do or how ants build their nests. That's not physics. I mean, I have felt this myself. I've been in complex systems for 30 years, and it seemed not a very well-seen discipline in physics. So particle physics is a serious discipline. Astrophysics is a serious discipline. It's complex systems, well, you know. Okay, so we had to wait until 2021, where for the first time a Nobel Prize in Physics was granted to a physicist working in complex systems. This is Giorgio Parisi. He received the prize together with two other people who had been working on climate change. One of them making the first models of climate change, the second one explaining that it was the anthropogenic effect that had a main role in the changes that we were seeing. And although difficult to understand, actually, if you go to the description that the Swiss Academy, the Swedish Academy, thank you, Borja, makes of the work of Parisi, it's important for people to understand. Right? Also, Parisi is difficult to understand himself. We have here Ugo Bastola, who was doing his PhD with him, with Giorgio Parisi. And, well, I don't know, you managed to understand him somehow, right? But he was difficult. Okay, but anyway, you see what he says here. A key task of the physics of complex systems, especially when applied to other fields, that I would say is mostly it's applied to other fields, is to identify universal quantities. This is what I was thinking of when I was saying that temperature is an emerging property that does not depend on the nature of the gas or the molecule that is involved, right? Measures that do not depend on the microscopic details, and universality classes, that is, groups of systems that exhibit the same behavior. That's also an important point. When you get rid of the details, sometimes you have a collective behavior that you can use in one or another system, regardless the microscopic structure of those systems. And this is something that I have done through my career in different systems. So just to more or less conclude this part, I very much recommend this paper, this article by Philippe Beau. He says, when physicists speak about the physics of a problem, they mean something close to what Aristotle meant. Neither a bare mathematical formalism, not a mere narrative, right? But a way of deriving process from fundamental principles. So when we work with biological systems, what we do first is to get inspired or to analyze data. So at the beginning of my career, it was more getting inspired. Now it's more going to data, because the amount of data available has increased so much. since in the last decades, right? So you go to data, you analyze those data, and you try to find patterns. What do I see here? Is there any kind of organization of regularity that I can extract from this data? When I have this, I have to know about the system to synthesize the main processes that may be shaping those data and the behavior I see that is communicated by this data. When I have the process, there is a further synthesis that sometimes is possible, sometimes is not, which is to translate these processes, these mechanisms, into a mathematical formalism. Sometimes we can do it, sometimes the system by itself is so complex that we stop here and we resort to simulations, for instance, to computational simulations of what the system is doing. Protein folding, for instance. I cannot write a closed mathematical equation for protein folding, but I know enough about interactions such that I can simulate the process, right? And more with AlphaFold now that we don't know how it's working, but anyway. Then once we have this modeling, we derive results, and we usually derive predictions, high-level predictions, like this temperature, right? How should the system be behaving? A good model should tell you something that you have not measured in your system, and then you go back to checking if this model, what this model is saying is something that can be falsified with data, right? And so there is a sort of circle when we do these approaches. The final product is hopefully we learn something about the basic mechanisms governing the system, right? So I'm going to make now a detour to tell you a little bit about my trajectory. Why do I put this image here? Because as a kid, I wanted to be a naturalist, a 19th century wealthy man naturalist, right? Which is something that, of course, by the time I was born, my sex and my familial condition was something not possible anymore. But this was my dream. So having a wonders cabinet at home, right? So I was collecting everything in the garden, and I had butterflies, I had worms, I had, so my socks and everything was put somewhere else, and I had all this sort of stuff trying to collect them. So since the Wonders Cabinet didn't come to me, I decided to go to the Wonders Cabinet, right? So this is one of the reasons that I am here now, so somehow to close the circle of things that have wondered me ever since, right? Okay, so I was also fascinated by stars. So this is why I decided to study physics, because I thought, well, okay, I'll be an astronomer and blah, blah. So, well, you see, life decides. I did my PhD, and something else that I also wanted to highlight with this, with describing this trajectory is how I've been always out of place, right? So then you can understand that I'm more in place here than I've been everywhere else. So I did my PhD at the Department of Nuclear Physics and Engineering, right? But my supervisor was both a physicist and a biologist. And the problems we were tackling had to do, for instance, with the organization of tropical rainforests. So you certainly know Barro Colorado Island, which is a wonderful place in the Panama Channel. I had the chance of being there, just because I was doing these models of pattern formation in rainforest. Amazing, right? So, well, we were looking at this data, which had been collected through many years on the island. And this was referring to the height of the canopy in different places, so higher or lower, less or well, cutting at 10 meters, right? So we saw these patterns, and this is not a random pattern. This is not homogeneous. You have some clusters, and you have some structures, and then we decided to analyze it with the tools of physics and maths. So we can calculate correlation functions between pairs of points and different things, and we proposed a cellular automaton model, which was very nice. And actually, this paper is one of my most cited papers. So this is one of the, I was saying, I wanted to give some examples of models I've been working in that I feel should be close to some of the science you do, right? The second project that I had for my PhD was the analysis of the temporal fluctuations and the correlations in the fossil record, right? So we had this data, and at the time, it was, so physicists were starting to look at data. This was relatively simple data. It's just somehow naive when I look back to this paper. It's 30 years by now, and it was, I don't know, something very innocent in a way, right? But what the viewpoint was, again, trying to make sense of these distributions that actually do not divide the extinction events in the big five and the rest, or the background extinctions and so on. You have a continuous distribution of extinction sizes through the fossil record, and then when a physicist sees a continuous distribution, this somehow means that there should be a unique mechanism explaining all this distribution and not separating one from another. And this is what we did. We had the model for that, and we managed to publish this in Nature. That's incredible. This is my first Nature paper and my last, right? So, and it was quite, so again, with the eyes of today, it's relatively simple what we did. But somehow conceptually was a kick to an establishment that didn't think in this way, in these terms, in these systems, right? And it was done in collaboration with Michael Benton, who was the compiler of the fossil record at the University of Bristol, and Per Bak, who was the father of what is known as self-organized criticality, which is this idea that you have these emerging properties not through external fields or mechanisms, but through an internal organization of systems, right? So then I moved for my postdoc, again, to an institute for chemical physics and electrochemistry, where I was doing also something that didn't have much to do with the institute, which was genealogy. Yeah, so you take a random individual, right? And you look at the ancestry of this individual. And something that you know is that this has been repeated, so this is the paradox of the grandfather. Right? You have 2 fathers, 4 grandfathers, and so on, and you multiply by 2 your ancestry. But it turns out that in a few centuries, the number of your ancestor is as large as the population in the world at that time. So it cannot continue to grow, right? So what happens here? Well, the fact is that actually, as you know, you have long-range endogamy, right? So you have some examples of people who are two sisters that married 2 brothers. These are close examples. But also it's very often that you have, oh yeah, but because someone, so back 3 generations with a second cousin of someone else. So this is something that occurs continuously, and this means that you have many repeated ancestors in your trees. So what we tried to do is somehow thinking also of monoparental trees, which is what you have in phylogeny and in the coalescent that is used usually for sequences. If you have biparental trees, the statistical properties change very much, right? You have this repetition. You can find, for instance, that the most recent common ancestor is not something that is linear in the population size, but logarithmic in the population size. So it's much closer than you may think. And, well, we had our paper published. Then the paradox was solved. Actually, it's not a very much cited paper because there wasn't much else to do afterwards, right? And there is a very nice, if you're interested, a nice paper in the American Scientists. They were very helpful in writing the paper, talking about these facts of genealogy in the area in genomics. It's illustrated also with royal ancestries. This is this was representing the Wars of the Roses, where you had these wars for the throne of England at the time. And it's fun because all these people were descendants of Edward III. But if you look at the royal houses of Europe nowadays, all of them are descendants of Edward III. And if you look at the whole population of England, about 80% of the population is descendant of Edward III. So probably you too, Rob. Yeah, you also have a royal ancestry. Which doesn't mean anything because you have also lots of thieves of the time and so on, like everybody else. Okay, so this was a fun problem to work with, and it was related also to this other, to the inheritance of culture that I also face it later. This was my second postdoc in Germany, Max Planck Institute for Colloids and Interfaces. There I was doing ecology, yeah? So this was a brief stay, about over a year, and we were dealing with the shape of ecological networks and trying to see how through local-Volterra equations you could construct or you could make sense of the structure that these networks had. At the time, again, it was simple data. It was slightly before all this boom of mutualistic networks that came later and so on. So this was sort of, okay. Then I came back to Spain to the Center for Astrobiologists, and I became an astrobiologist. Okay. So I was there for 13 years, actually. And this is, so there were two main lines, and this is when I started also what is my main line up to today, which has to do with molecular evolution. But I also did some things about cultural evolution, just thinking of, of a process that, again, is not only for culture, but it can be also applied to populations, to heterogeneous populations. When you have this, say, coalescent with many different groups, right? And you go back to the common ancestor, but you also have an introduction of diversity through mutations of any type, then you have populations that are in a stationary state that is heterogeneous, highly heterogeneous. And this is the case of viruses, and is the case also, for instance, of family names, which is one of the problems that I was working with. So this is an old problem that goes back to Galton, actually, of trying to understand why you have some names getting extinct and some other names that have Garcia, that many people in the population have, right? So this is, you can make sense of that. This is a multiplicative process, and we did some studies on that with data, with more data. And also in the area of cultural evolution, this is how languages are propagated, say, are inherited from your, one of your parents. Actually, in general, if you consider it to be a monoparental language. And okay, there was a lot done about that, but we were interested also in what was determining linguistic diversity. And it's very interesting because the patterns of linguistic diversity are quantitatively equivalent to the patterns of biodiversity if you look at latitude. Right? You go to the north, you go to the south. And also other quantities that we measured, for instance, the area where a language is spoken, and you compare with the number of speakers of the language, is equal to the distribution of areas that the species is found in compared to the number of individuals of that species. So there is something self-organized that has not much to do with whether a language is thought to be better or worse in communication, which is not the case, right? So it can be put in correspondence. I have also something written about that, this correspondence between biological diversity and linguistic diversity. And it's nice because already Darwin knew about this universality, right? He writes in The Origin, the formation of different languages and of distinct species and the proofs that both have been developed through a gradual process are curiously parallel. So he had already this intuition. Right? It's just an anecdote. So linguistic evolution was banned for almost a century by the Royal Society. It was forbidden to study linguistic evolution. So the reasons, who knows? Okay, and then here I started actually these studies on molecular evolution. I started with viruses and the RNA world. And I was interested also, so you know this, I started with macroevolution and went to ecology, and I thought, well, evolution also starts at lower levels. Sometimes I would like to go the other way up to the higher levels, but it's difficult, right, to try to find out how this molecular evolution gives you some ecological complexity and back to a global ecosystem. But anyway, so this is a sort of background image that I have. And so I have devoted much effort to this molecular evolution, trying to understand what are the dynamics of heterogeneous molecular populations and how the dynamics condition this adaptive ability. And I've been working a lot on genotype-phenotype maps at the molecular level, right? And a bit less on the structure of fitness landscapes. Then I moved to the National Biotechnology Center because I thought, oh, okay, people know a lot about molecular biology there, yeah, but they don't know much about evolution. Actually, they don't care much about evolution, which was something, well, yeah. So I worked a lot with this kind of objects, which are genotype networks. A genotype network essentially is the collection of all the genotypes that give you the same phenotype. You have to define phenotype. Phenotype can be, in the case of a protein, the 3D structure of the protein, you can put other features in the definition of phenotype. For RNA, it can be the folded secondary structure or whatever, right? And then you can enumerate, well, not always, because we are talking about very big numbers here, but you can, in principle, establish all the genotypes that give you the same phenotype, and you can study these objects as mathematical objects, as networks, right? And they have very interesting properties. So I have devoted much effort to this topic, and if you are very interested in it, right, there is this review that we wrote together with about 20 other researchers on the field. And it's a 50 pages paper review on the state of the art of going from genotypes to organisms, what we have learned computationally, what we have learned analytically, and which kind of empirical evidence we had at the time. And then I moved here, right? And here I'm continuing this research on evolution at the molecular level, especially with viruses. So so a few years ago, I thought, okay, we have these descriptions of these genotype networks, theoretical genotype networks, but actual populations are very far from being able to navigate a full genotype network or to explore a substantial part of these networks. And I'll give you some figures in a moment. So what we know is that this sequence evolution occurs on sequence spaces which are hyper-astronomically large. I don't have an adjective for these sizes. Well, okay, any typical genotype network for any modest length has more genotypes than particles are in the universe. So there is no possibility that we will never, ever exhaustively enumerate these genotypes or find any information from a computational viewpoint, right? But actual populations do not explore all these possibilities, right? Actually, they are very locally confined. And the proof of that is that you can do phylogeny and go back 3,000 million years back in time, and you can find still similarity between sequences, right? So, I mean, it has to be very local. So, phenotypes, viable phenotypes, and adaptive phenotypes are very close to any random sequence, because otherwise we would not see the complexity we see, keeping memory of where we started, right? So we decided to explore this question with the help of a phage, the Q-beta phage that infects E. coli, and a colleague of mine, Esther Lázaro, at the Center for Astrobiology. She's doing in vitro experiments with this virus. Okay, and we said, well, let's start with something that we can manage. Let's take an amplicon of the virus. Actually, we have various amplicons, but say this one, right? Deep sequence the virus and see what is the diversity that you have there. And again, some figures. The total number, this amplicon has length 271. Total number of different sequences of this length is 10 to the power of 163, right? The number of virus particles, any kind of virus, on Earth is estimated at 10 to the 31. The number of particles in the universe is 10 to the 80 at most, right? So we are so far, even with a single amplicon, from exploring a substantial portion of a space of sequences, right? So these were our questions now. So how is a viral quasi-species organized in sequence space? To what extent does this organization, the network that this population, the genomes of those populations might form, the genotype network, resemble our expectations from theoretical models, right, from theoretical GP maps? And whether these neutral networks are essential for navigation and adaptation. You may know, well, there was Kimura's neutral theory, and there was also afterwards Maynard-Smith suggesting that you have these neutral networks that percolate the space of sequences, so you can make point mutations that are neutral and actually explore all the neighborhood of these point mutations. So you move step by step and you explore close neighborhoods, right, of each of those mutations. And this solves the problem of typical, well, it was Salisbury a year before Maynard-Smith proposed this case that said that it's impossible to have function because because of these numbers. And if you have to go to a specific sequence that produces a specific function, then of course there is no chance if you have to do it randomly, right? But Meyer Smith said, no, okay, you can navigate these spaces and you can explore many alternatives because many of the changes are neutral. So our question was, are these neutral networks essential? How far from where I started am I found after adapting to the different environments that a species or a population has seen through geological time, right? Okay, so this is not that ambitious. So we were studying the evolution of these viruses for 60 passages in the lab and forcing the virus to adapt to different temperatures, right? At the end point here, so the experimentalists took these populations, took samples, and sequenced, as I was saying, the amplicon, one of the amplicons of this virus. And then we reconstructed the networks, right? Networks are reconstructed in this way. You have your sequences, your abusing language, we call them haplotypes because they are all the sequences that are identical. So this is a haplotype, this is a node of my network, and two haplotypes are connected if they are at the distance of one mutation, right? So this I do for one of the final populations in the final stages, and I also do it for the three populations at the end of this evolution experiment and try to see how they organize one to another, right? Okay, so the first thing is that the numbers we get for the diversity of these viruses is huge. I mean, sometimes I see people talking about polymorphisms, but viruses are really orders of magnitude above that. We have on the tens of thousands, 10,000, about 10,000 different sequences, different haplotypes in each of these populations, which is a little bottle in the laboratory, right? And well, these are the edges, so the number of links I have among those populations. When I represent these populations, I see that they have this kind of structure. So I'm taking the most abundant haplotype, the one represented by the largest number of sequences, and I put in concentric circles here mutants at distance 1, differing just in one position, in two positions, in three positions, and so on, right? And this is the structure we see. Today, I'm not going to present the analysis on the structure of these networks, which I think is interesting because we have topological properties that can be put in correspondence with selection, this structure is repeated also in the three viruses with essentially the same statistical properties in the three end populations. So they repeat this structure, and when we quantify this structure, there is no difference among the three populations, despite the fact that they have fixed adaptive mutations. So the population at 30 degrees has fixed one mutation with respect to the consensus sequence of the original population. Population at 37 degrees has fixed one mutation, different mutation, although it was evolving in the same environment than the initial population. And the population at 43 degrees has fixed two mutations. But even if these mutations are fixed, the population displaces itself and locates around the new fixed mutations. We see little trace of the movement of the sweep between the population being at one place and moving to the other place. And this hierarchy in the searching process is very rapidly reconstructed. Right? So making some numbers, what we see is that essentially the populations explore all mutations up to distance 3. All mutations. No selection, no cherry picking. I mean, it's a brute force local exploration of sequence space. You see that the abundances at distance 1, distance 2 are different, right? Some of them are more abundant, some others are less abundant, but still they are represented. And maybe some of them are lethal, but there is this brute production of variation through mutation that we believe is the core of how viruses search sequence space and go through valleys. You have these ideas of valleys, of right landscapes, right? But actually, here we have many sequences which have very low fitness. We suppose they have very low fitness because they are not that abundant, but the population tunnels through those sequences by producing a large amount through mutation. Right? So what we see here is that the landscape is non-neutral. Of course, there are differences in fitness, but that there are multiple phenotypes represented. I'm not showing here today the network of different proteins, but actually what is actually neutral here is a small fraction of all these mutations. And you have many others that, I had a student looking at distance one, how many substitutions in the proteins, these are coding parts of the virus, how many proteins you have, and essentially you have all possible amino acids at distance one. So it's saying everything at once, this virus. It's also very much out of equilibrium, so you have this punctuation, say, you fix a mutation, reconstruct search process, fix another mutation, reconstruct again the search process, and so on. So there is this very maintained profile of search, but at the same time, you have this out of equilibrium behavior where you jump from one mutation to another if they are better than what you have at hand, right? Okay. So there is this memory also of the initial condition that is essentially what allows you to make phylogeny, because you can trace back the original population when you look at the populations you have at present, right? This is a representation of the aggregated network. Here we have represented all genotypes. Again, these circles are the original most abundant sequence in the ancestral population, and the most abundant sequence at 43 degrees, 2 mutations away from this one, and same for the other two populations, 1 mutation away. And you see that there is sort of displacement in the space of sequences. So adaptation, there's this ecological speciation, right, in my system due to these different environments around. This is a different representation where we have collapsed at each of the vertices of the triangle all the sequences that only appear at each of the environments. And what you see is that, okay, there is a fraction of sequences only appearing at each of them, but there is also a large interior of this triangle of sequences that are present in all 3 environments. So there is a high degree, at least at these time scales, of pre-adaptation to the environment. Sequences that were present at 37 degrees also can do relatively well at 43 or 30 degrees. And then you can, from there, from half an eye, you can start adapting to the new environment. Right? Okay, so this is another representation of this high level of pre-adaptation where I here only have population at 30 degrees, population at 37 degrees, and the population at 43 degrees. And you see that also the other networks are populated at each temperature. Right. Okay, so this is more universal than we thought. It's not only for Q beta. We also had some intra-patient samples of SARS-CoV-2, and the structure is very similar. And if you look, for instance, on how the space is explored, the difference between, these are the Q beta samples, the 3 upper lines, and these are the SARS-CoV-2 samples, and they are slightly below because the coverage of the sequencing was lower. So if you increase coverage, you move the system to explore, to better explore and hierarchically explore the surroundings. Okay, so we think this is a very general pattern, and now we are trying to get data from DNA viruses also, which virologists think they behave differently because the mutation rates are relatively smaller, but from a physics of complex systems viewpoint, they should behave probably similar, right? So let's see what we get. Okay, these are my acknowledgements for the people with whom I've been collaborating in the last decade or so. There were many more if I would go to the timeline I have shown you for the different centers where I was. And well, Samuel is here, Iker is here also at the museum. And I keep in touch with all of them essentially because they are involved in this project. Before finishing, I want to make 2 announcements. First, since January, I'm a member of the board of the Philosophical Transactions of the Royal Society B. So I would be happy if any of you wants to edit a volume on any topic. You can come to me and I can give you some information or about the process and so on, right? So happy to help if you find it interesting. And something not that serious, I have a podcast with some friends and colleagues, which is called The Spherical Cow. La vaca esférica. These are my colleagues. You more or less recognize us here. And so if you want to know about not only physics, but also many other topics, this might be your podcast, only maybe, yeah? Okay, so, but this is something that we are more like a jazz band than a group of physicists, because we play wherever comes in the day. So you have lots of chapters about different things. Yes, there are physics, but there is also psychopaths, microbiome, language genealogy, time travel, and other stuff. So, and with this, yes, I finish. I'm very happy to be at the museum. I'm looking forward. I have a coffee machine in my office. You're always welcome for a chat or for a coffee. Yeah.
Okay, Susanna, thanks for trying to make simple, complex physics, biological systems, and emerging properties. Time for questions. Who starts? Juanica.
Thanks, Susanna. Thanks, Susanna. It was a really inspiring talk. I have two questions. The first one about the genotype networks. So as a biologist, to me, it's a bit difficult to digest this thing that you said that every sequence is tried by the viruses. I'm not questioning what you said, but it's a bit difficult to digest
Data,
this.
yeah.
But from the size of the circles, it seems that some of the genotypes are more frequent than others.
Absolutely.
But I would think that, for example, synonymous mutations are not a problem. But at some point, a protein would lose the function. Maybe it's not,
Right.
but this can happen in a protein that is, I was going to say, that can be lethal, but anyway, viruses are not alive. So I don't know, what do you say about this?
Okay, so the first thing is that I don't want to claim that what viruses do, other organisms do, cellular organisms do. So first thing, because these populations you have, there are a lot of interactions between the products, the translated product of all the sequences, right? So if there are proteins that do not work, but you have a fraction of proteins that do work, there are other sequences that are using these viable products even to replicate themselves, right? So you have an ongoing process of collaboration and competition within the viral population, right? Now, the most abundant sequence, of course, plays a role. I mean, this has to be functional in a sense, right? But the point is that it's so abundant and the mutation rate is so high that you're producing everything 1-2 mutations away from this source, right, from this sequence. So probably you have purifying selection that cleans many of these variants. And you are there searching until you find something that is better, and then you move to this new center, and you repeat the process. It's just the mechanism of searching that is, say, represented by this data. Is not strictly selection. You're not the referee, right? Okay, so because we had a long discussion about where the signatures of selection are seen in those data. And actually, you see the signatures of selection at the first layer just because abundances change, right? But not because presence of variation is selected. And you see purifying selection in the periphery, where then you have your haplotypes are rare, they don't have the strength to populate the surroundings, and then it's like a sink, it disappears, right? So this is our understanding of the process right now.
Anyway, I thought it was very interesting that this is you can see what we always say, but we never see that mutation is random. We say this, but we don't see it, no?
Yeah.
Second
well,
question.
There could be many other things that you could do, because I know that sometimes you have biases in mutations that are present or not. And this is something that we have not tested. We have measured the NDS ratios, for instance, for all the sequences, but you know it's not that meaningful when you have mutationally coupled populations. So you have to be careful with your conclusions about that. right? So, and there are many other probably analyses that could be done. The data set is very interesting. You have also tens of populations, hundreds. So,
Cool. The second question is about something you said about looking for patterns into data. I understand from your talk that you are not one of these people that say that science should be hypothesis-based, but this is something that I heard many times, mostly from people working in the lab, in the wet lab.
Yeah.
To me, this dichotomy between looking for patterns so like data science and hypothesis-driven is a stupid split, but I don't know. What is your opinion on this?
Well, I mean, you have to combine your educated guesses, which is you try to look for something, with your prejudices, because you come from a field that has taught you that there is purifying selection, or that the synonymous mutations matter and the others do not. And I mean, or that you can visualize things using a fitness landscape that is a real landscape, topographic landscape. I mean, I think that in this direction, collaboration is the best way. So I think that different disciplines give you a different view of the same problem, and each of them has their own, say, assumptions, but there are different assumptions, and then I think that this combination is okay. So hypothesis-driven has the problem that you might be affected by the dogmas or the things that you believe to be true within your school of thought, right? And data-based sometimes happens that you have lots of data, but you don't know what to look at, right? So I very much dislike these approaches where you just take the data and you put it in a black box and you get something out, because I do not gain understanding by doing that. So, I mean, yeah, we do our best.
Cool, thank you.
Welcome. Hi.
Thanks for the talk. I have a more generic question, which is related to, I think, for most of us, your amazing career so far. You went through different, I mean, I'm talking about the fields that you went from rainforests to sequencing in the molecular world. So my question would be related to scaling. And do you think that going from the universe of the molecular world, where we have uncertainties across genes and across subspecies and then species, do you think that there is a simplification in the structures as we go towards biomes, for example, in life? Do you see any similarities across these different universes of scaling, or do you see that life holds the same components? I know that it's a huge question to ask, but I want your opinion on that.
Say it again. I missed something you said. When you go from molecular?
When you go from the world of the molecular world to the world of biomes, because you work with both.
Yeah. It's obviously a very difficult question, but in biology, my understanding of how higher levels in biology are organized. So you have, look, sometimes in physics, when you study a gas, you have this microscopic description, and then you can extract temperature. That was statistical physics does, right? But in biology, once you have something that is a little bit more complex in the upper level, you have a feedback to the lower level. So all the levels in biology are coupled. I mean, reductionism is not a good approach for biology. You start microscopically, but sometimes you cannot make sense of what is happening microscopically if you don't think of the ecology. And I can give you an example. I was working for some time with multipartite viruses, which are fascinating entities. They encapsulate their genome separated in different parts, and they have to co-infect the host in order for the genome to be together. And so, I mean, there is no clear... Until today, there is no clear understanding of why this is a winning strategy for viruses. But we had a proposal, and only if you include ecology can you explain why this can be a winning strategy. And this is because when you combine parts, and if you think of viruses and satellites that may co-infect or may not co-infect, right? Co-infection changes the pathology of the disease, of the effects it produces. And it's very easy, if you have all sorts of fragments, to co-infect plants in particular, because they have many viruses, so infecting at the same time. And then, if you have a new host, instead of trying to adapt by mutating, I just pair with someone else, and I can be fast. First come, first served. And this can be a winning strategy, but only if you think of the ecology. So microscopic analysis is not enough in this case, for instance. I can't tell you much more examples so specific, but I believe that you always have to keep in mind this feedback between levels, because otherwise you cannot make sense of some things that we observe. Right?
I have always enjoyed a lot your talks.
Always. Thank you. He said he has always enjoyed my talks.
Sorry. Yes, I always enjoy your talks. I knew you at the CMB, and well, always is amazing. Feynman usually said that there are pretty much things that can be known, that those that can be demonstrated.
Right.
Your approach, your seek of patterns are always very funny, but you have been to face this assessment very often, I know, I think. How do you face that in the case, for example, of analyzing emerging properties that usually are going to be affected by spurious correlations because you are detecting them at a time, and it's very suitable to think that some of them are related, but you cannot get the proof of that association. How do you work with that kind of assessment?
Let me rephrase you, see if I understand you right. You're talking about the spandrels? So things that are not selected or that do not have a main role, a primary role, but are there. And I might try to make an understanding of those structures, for instance?
An example, for example.
Okay.
In the extinction paper, you identify a common pattern, a common rate of extinctions. But how do you know, well, it's pretty related, but how do you discriminate among the influence over the stratification process of the geological processes that you are using to infer that climate changes and to relate that with extinction events? Because at last, you are identifying only the ability of the soil to preserve those specimens that you are recording to analyze extinctions.
Yeah.
So it's a potentially spurious correlation. But of course, that makes pretty sense. That makes sense.
Okay,
That
okay.
makes sense.
So first
that kind of
okay.
Thank you.
First a comment about data. Data from any system, from any experiment, is already conditioned and biased by our preferences and by our technology. So I can do deep sequencing, I do deep sequence, right? And there are other things, but I need some primers. Primers are selecting, right? Well, but this is my best now. Okay, so I mean, this is also something that we have to keep in mind. Also for physics, you have some instruments that measure some quantities and not some others, right? And you try to make sense of what you see through your lenses. I mean, we cannot escape that, right? Then, that said, there are ways of trying to avoid that the results you extract are so much conditioned by the way in which you have obtained data. For instance, in the fossil record, for different organisms, you have different characteristic times. Your box time, typically in those representations, was about 1 million years. Sometimes it's 5 million years. And for some marine organisms, which are well preserved, you have smaller scales, right? The essence of our work was that there was a self-similar pattern. So it doesn't matter whether my grouping is changing, I see the same kind of behavior. And one of the main results was that correlations in the extinction patterns extend far beyond the time, the lifetime of genera, right? So, I mean, this is something that transcends the details of the data. But of course, you have to always be careful about that. But I mean, to keep in mind, it's not just, yeah, sure, but, yeah.
Thanks.
Yeah.
I end with a thought, is do emerging properties explain bureaucracy or not?
Well, I mean, I
From
think...
simple things you get to very complex.
Yeah, there are papers on that, but I don't think you want to read the literature.
Okay, thanks for coming. We might
Thank you.
go, despite it's raining and there are many problems, we might go to
Yeah.
the kiosk for some beers. Thank you.
Thank you very much.